Hi Emma, could you explain what a model organism is and why it's so important in research?
Certainly. A model organism is one that researchers study intensively to understand biological principles that can apply to other organisms as well. These organisms often have specific traits that make them ideal for research. They typically have short life cycles, produce large numbers of offspring, are economical to maintain, and have fully sequenced genomes. These characteristics allow us to generalize findings across different species. Examples of model organisms include bacteria, yeast, frogs, mice, and even plants like corn.
Interesting. Now, what about tissue and cell cultures? How are they used in research?
Tissue and cell cultures allow us to study human cells in a controlled environment without needing a full organism, which is essential for ethical reasons. This approach, known as in vitro, involves cultivating cells outside their natural environment under precisely defined conditions. We use these cultures for various purposes, including drug development, genetic studies, and even prenatal diagnostics.
And what makes this method advantageous?
Using cell cultures has multiple benefits. It allows us to get results quickly and control every factor in the cell's environment, which isn't possible in a whole organism. Additionally, since these cells are outside the body, we don't face the ethical issues of experimenting directly on humans. Cultures are also quite accessible for genetic manipulation.
Are there any downsides?
Yes, there are. Cells in a culture lose the context of the whole organism, so we miss complex interactions that happen within a living body. Additionally, there's a risk of contamination, and over time, cultured cells can accumulate mutations, altering their properties.
That makes sense. So, how do you set up a cell culture?
When we start a culture, we need to transfer tissue into a nutrient solution as quickly as possible to mimic the body's environment. We control various factors like salt concentration, pH, and temperature. A laminar flow cabinet sterilizes the workspace to prevent contamination. We use a growth medium, usually with serum, nutrients, and growth factors, to sustain the cells. Equipment like CO2 incubators, inverted microscopes, and centrifuges are also essential for observing and maintaining the culture.
What are the main types of cell and tissue cultures?
There are two main categories: tissue cultures, where cells remain attached and interact, and cell cultures, where cells are isolated. Primary cultures come directly from an organ and contain a mix of cell types. They don't survive long but can be passed or passaged into a new medium. Secondary cultures are derived from primary cultures and can survive through multiple passages, though they still have a limited lifespan due to the Hayflick limit.
What about cell lines? I've heard that some cells can divide indefinitely.
Right, cell lines are what we call immortal because they can divide without limit. This can occur naturally, as in cancer cells, or by introducing specific mutations. A famous example is the HeLa cell line, derived from a cervical cancer patient named Henrietta Lacks in 1951. HeLa cells have been crucial in developing the polio vaccine, cancer research, and even space experiments.
Speaking of cell growth, what are the stages cells go through in culture?
Cells in culture progress through several growth stages. Initially, there's a lag phase, where they adapt to the new environment. This is followed by the log phase, where they grow exponentially. In the stationary phase, growth halts due to nutrient depletion and toxin accumulation. Finally, if conditions aren't improved, they enter the death phase, where cells start to die off.
I've read about stem cells, too. Could you describe their role in research and medicine?
Certainly. Stem cells are unique because they're undifferentiated, meaning they can develop into various cell types. Embryonic stem cells are pluripotent, capable of becoming almost any cell type in the body, while adult stem cells, like those from bone marrow, are multipotent and more limited. We use stem cells for regenerative medicine, repairing damaged tissues, treating neurodegenerative diseases, or even reconstructing organs.
That's fascinating. And is there a special material that's used to grow cells in 3 dimensions?
Yes, that would be matrigel. It's a protein-rich, gelatinous substance secreted by mouse tumor cells, used to mimic the extracellular environment. At low temperatures, it's viscous, but at body temperature, It forms a gel that allows cells to grow in 3D, simulating conditions in a living organism. It's invaluable for studying cancer, stem cell differentiation, and drug testing.
Thank you. This really clarifies how cell and tissue cultures are used in research and medicine. What does it mean to passage cells in culture? Is it a common process?
Yes, passaging is essential for keeping cell cultures viable over time. As cells grow and cover about 70% of the culture dish, they use up nutrients and space. We then transfer some of these cells to a fresh medium in a new dish. This prevents overcrowding, called confluence, which would otherwise halt cell division and lead to cell death if not managed.
Are there special steps involved in passaging?
There are. First, we remove the old medium, wash the cells with a phosphate-buffered saline solution to remove residual nutrients, and add an enzyme called trypsin. Trypsin detaches the cells from the dish, allowing us to collect and transfer them. We then centrifuge them, removing any enzyme remnants, and resuspend them in fresh medium.
I've heard cells can be frozen for long-term storage. How does that work?
Correct. This process is called cryopreservation. We cool the cells very slowly, often using controlled-rate freezers to prevent ice crystal formation that could damage the cells. A substance called dimethyl sulfoxide, DMSO, acts as a cryoprotectant, preventing crystallization. Once frozen, the cells are stored in liquid nitrogen. When needed, we thaw them quickly, reintroducing them to the culture under optimal conditions.
That's incredible. So, what about the growth cycles you mentioned earlier?
Cells follow specific growth phases. Initially, in the lag phase, they adapt to the new environment. Then comes the log phase, or exponential growth, where they rapidly multiply, consuming nutrients and producing byproducts. Once these nutrients deplete, the cells reach the stationary phase where growth stops. Without intervention, they enter the death phase, where cells start to die as the conditions become inhospitable.
Are there any specific tools or equipment required to handle these cell cultures?
Absolutely. Besides the growth medium and controlled environment, essential tools include a laminar flow cabinet to maintain sterility, an incubator to control CO2 and temperature, an inverted microscope for detailed observation, and a centrifuge for processing cells. These tools help create and maintain the ideal conditions for cell survival and growth.
Understood. Now, what's the role of stem cells in research?
Stem cells are key players in regenerative medicine due to their ability to differentiate into various cell types. Embryonic stem cells, derived from early embryos, are pluripotent, meaning they can become any cell type in the body. Adult stem cells, found in organs like bone marrow, are multipotent and are more limited, generally forming cell types within their tissue of origin. We're exploring their applications for repairing tissues, treating neurological conditions, and rebuilding damaged organs.
Is there anything unique about embryonic stem cells?
Embryonic stem cells are indeed unique because of their pluripotent capacity. They're often derived from early-stage embryos created via in vitro fertilization, where unused embryos are either stored, discarded, or sometimes used for research. These cells offer immense potential for therapeutic cloning and regenerative treatments, although ethical considerations guide their use.
And what are totipotent, pluripotent, and multipotent stem cells?
Good question. Totipotent stem cells, found in early embryos, can develop into a complete organism, forming any cell type, including those in the placenta. Pluripotent cells can develop into almost any cell in the body, but can't form an entire organism. Multipotent cells, like those in adult tissues, have a more limited scope, differentiating only into cell types within a specific tissue or organ.
Lastly, I'm curious, what challenges do researchers face with embryonic stem cells?
With embryonic stem cells, there are challenges on multiple fronts. Ethical concerns often arise, given their source from early embryos. Also, controlling their differentiation precisely can be complex. However, when managed well, they're a powerful resource, enabling groundbreaking advances in treating various conditions and exploring therapeutic cloning.
This has been incredibly enlightening. Thank you for such a thorough overview of tissue cultures and stem cells.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.